Hello, can you hear me?
Yes, sir, we can hear you.
Oh, okay. See,
We'll
this,
start in 2 minutes.
okay, how many number of audience this platform can accommodate?
So we have 250 maximum. But
Okay,
since we have it live streamed on YouTube, other people can also watch it.
okay.
I think it's 9:30, so we can start. Professor Mazumder, you're ready, so we can start?
Yes, I'm ready.
Okay, perfect. Welcome all to the Progress and Prospects in Biology webinar series. Thank you once again for joining from all around the globe. We truly appreciate your support and kind cooperation to bring the webinar series to you. A couple of housekeeping things to remember before we start today's session. Please mute your microphone, turn off your video, and don't share your screen in the Google Meet app during the webinar to preserve the bandwidth. Those who are watching from Google Meet, you can pin the speaker screen so you have an uninterrupted streaming of the slides. You can post your questions for the speaker using the chat box in Google Meet or on YouTube, and we will moderate towards the end of the session. Any questions that are not related to today's talk, please feel free to contact us by email. We share the feedback link to get e-certificates only at the end of the session, and it's available to all participants watching in Google Meet and YouTube. And no links will be shared by email. Even if you don't need a certificate, please drop your feedback, because we go through all the feedbacks and we try to bring them to you all. With that note, I'd now request Professor Enarai Banerjee to give the welcome address and start today's session. Thank you. Over to you, ma'am.
Thank you, Shaunak. A very warm welcome to our speaker, Professor Barsanjit Mazumder, and a very warm welcome to all the participants in today's talk, which promises to be very, very interesting. Not only people who are tuning in today in the Google Meet and in the YouTube, but also all the future participants who will be watching Professor Mazumdar's talk, which we shall archive in the YouTube channel. So a very warm welcome. Today's talk promises to be very exciting, not only because Professor Mazumder is a well-known scientist in the field of inflammation and viral inflammation. Am I correct, Professor Mazumder?
Yes.
Yes. So you will be hearing from the expert, and it makes all the more interesting, because as we speak, Professor Mazumder has himself volunteered for the second dose of the COVID vaccine, which is being made. While we speak, all kinds of immune reactions are going on in Professor Mazumdar's body, and we will be getting a glimpse of the molecular mechanisms that he has unraveled, he and his team have unraveled through his work. Professor Barsanjit Mazumdar is currently a professor of molecular genetics at the Center for Gene Regulation in Health and Disease in the Department of Biological Science at the Cleveland State University, USA. He did his BSc in chemistry from the University of Burdwan in 1979, his MSc in organic chemistry from the Vishwabharati University in 1982, his MPhil in applied chemistry from the Indian School of Mines in 1984, and his PhD in biochemistry from Bose Institute in 1989. He held the post of scientific officer in the geochemistry division of the Geological Survey of India from 1989 to 1991. He was a postdoctoral fellow at the University of Minnesota from 1991 to 92, and then at the Cleveland Clinic Foundation Lerner Research Institute from 92 to 94, after which he was a research associate at the same institute from 1994 till 2001. Professor Mazumder became a project scientist in the Department of Cell Biology at the Cleveland Clinic Foundation Lerner Research Institute through 2001 to 2004. After that, he became an assistant professor from 2008 to 2013 and a professor since 2013 at the Center for Gene Regulation in Health and Disease in the Department of Biological Science at the Cleveland State University. He is also an adjunct faculty at the Department of Cellular and Molecular Medicine of the Cleveland Clinic Foundation Lerner Research Institute and Lerner College of Medicine, Cleveland, Ohio, and at the RNA Center, Case Western Reserve University School of Medicine, Cleveland, Ohio. Professor Mazumdar's current research interest lies in the molecular mechanism of inflammation and resolution of inflammation, translational regulation of inflammatory gene expression in monocytes and macrophages, post-transcriptional regulation as a mechanism of resolution of inflammation, defects in the resolution of inflammation in cardiovascular diseases, mechanism of ribosome biogenesis and general mechanism of translational control of gene expression, and role of ribosomal protein in embryonic development. He has received several awards and grants and is a reviewer of several scientific journals and has published over 40 research articles in peer-reviewed journals. Today, the talk entitled GAIT, Gateway of Inflammation and Pathogenesis. So without wasting more time, and I venture to say that when I was reading your introductory CV, Professor Mazumder, I must appreciate the enormous different subject positions that you have held. And for our audience, this is very important because it also teaches you to look at a problem today. And over to you, Professor Mazumder.
Hello, good evening, everybody. Although here it is morning, and thank you, Dr. Ena Banerjee, for giving this very excellent introduction. So nice of you for your kind words. So thank you, and let's start. So I'm going to talk today on the center of my gravity, which is inflammation and pathogenesis. So inflammation, the term inflammation is pretty much known to you now, I believe. And what is inflammation? It is actually a double-edged sword. So we are being continuously challenged by exogenous pathogen, no matter where we are. So, but how we are not getting infected? So because we have a weapon system of our innate immune machinery. So that weapon system is being engaged continuously engaged with war with the invading microorganism, and actually most of the time it is defeating them. How? So because this lethal weapon system is very much efficient, and rapid generation of cytokines and other lethal molecules are a very important component of this weaponry. So since this is a war and you are using your lethal weapons, and you better know how to control them. So if you don't know how to properly use them or control them, most likely that the effect could be counterproductive. And failure to control those weapons, could actually, instead of killing your enemies, might kill you or your friend or your own home. So it is absolutely necessary to control these lethal machine. So how cell has evolved to do that? You might see a lot of work has done on how inflammation activates, like TLR pathway, NF-kappa B pathway. However, how inflammation resolved, there are still the knowledge is evolving and limiting. So we discovered one such mechanism, and I'm going to take you through that saga today. So what we have found, this is a story, this story has emerged maybe several decades ago. So when I was in the beginning of my career in Cleveland Clinic Foundation. So what we have found, that if you treat monocytes or macrophages with interferon gamma, it induces one inflammatory protein, ceruloplasmin. So that protein is highly inflammatory. It goes high in infection. But we have found a very weird activity of the cells. So when we treat the cells, monocytes and macrophages, with interferon gamma, so we can see that mRNA is induced, and we can see de novo protein synthesis by S35 methionine labeling. So I'm not going to the data per se, just showing you with model just for the sake of the time. So there are new protein synthesis, and that is not surprising because mRNA induced protein synthesis will happen. But what is surprising, between 16 to 24 hours of post-interferon gamma treatment, we can see a lot of mRNAs hanging around. However, there is no new protein synthesis. So what is happening there? So I pursued that and identified actually the mechanism how it is happening. So again, I'm summarizing lots of data with just one model, just for the sake of time, so that I can discuss some of the recent data. So what we have found, that upon treatment with interferon gamma, one protein got released from the large subunit of ribosome. So all of you, I believe all of you know what ribosome is. Ribosome is the protein synthesis machine of the cell, and ribosome is a complex of RNA and protein. And there are two subunits of ribosome. One is large subunit called 60S and small subunit called 40S. And all ribosome contains RNA and protein. So one of the protein of the large subunit is L13A. So what we have found, that if you treat cells with interferon gamma, this L13A become phosphorylated and then released from ribosome, and then released and phosphorylated L13A binds three other protein. It's called GluPro-RS. So this protein is one of the protein that involved in the amino acylation of tRNA. So GluPro-RS, NSAP1, and GAPDH, and form a multi-protein complex, and that complex binds a specific element harbors in the 3' UTR of its target message. So we name that element as GAIT element. What is GAIT? Means gamma activated inhibitor of translation element. So whatever mRNA harbors this element, this complex sees that as a target, binds the 3' UTR, and then inhibits translation. So at this point, I moved from Cleveland Clinic to Cleveland State University as a faculty and established my own lab. And one of my graduate student, Purvi Kapasi, who was the first graduate student in my lab, identified the molecular mechanism at protein to protein interaction level. What is actually happening? A protein can bind the 3' untranslated region of a message and silence translation. So since this is translation, I just want to give you a 101 of translation, that translation requires RNA protein interaction. So this, what you are looking at, the celluloplasmin ORF, see, all the mechanistic work has been done using celluloplasmin mRNA as a model, as a first target of this control activity. So this is the start codon, this is the stop codon, this is the ORF. And this part is the 5' untranslated region, this part is the 3' untranslated region. So in the very end of the 5' untranslated region, the message is capped. You know, most of the eukaryotic message is capped at the 5' end. And this capping recruits a complex called translation initiation complex. First binding, cap binding protein EIF4E, then 4G, then these two helicases, and then form the EIF4F translation initiation complex. So this is happening at the 5' extreme 5' end of the message, but at the extreme 3' end of the message, poly A binding protein binds the poly A, and this poly A binding protein, or PABP, also has a strong affinity towards 4G. So a protein at the head is binding a protein at the tail. This head-to-tail binding is bringing the translating mRNA as a closed loop shape, or closed loop translation model. And by virtue of this closed loop structure, anything that binds the 3' UTR, if an inhibitory complex binds the 3' UTR, that complex might get a access to the initiation complex and thereby inhibits translation initiation. So what Purvi has found, that L13A as a part of the GAIT complex, that means binding to the GAIT element, can directly interact, occupies the scaffold protein EIF4G, and do not allow the recruitment of the small ribosomal subunit. If recruitment of the small ribosomal subunit do not happen, then translation initiation do not happen. So this is the way the GAIT complex binding the 3' UTR is directly blocking translation initiation. And we published that paper a long time ago. So now such a sophisticated machinery for translation control may not have evolved only to silence one mRNA. That doesn't make any sense, right? So we hypothesize that maybe there are maybe a cohort of other target message. So we initiated a genome-wide search for the target mRNA for interferon gamma mediated and L13A dependent translational silencing. So the way we did is a very unbiased approach. So we treat the cells with interferon gamma, immune cells with interferon gamma, and then make lysate, and then resolved by the sucrose density gradient to resolve it with the translationally active pool of mRNA versus inactive pool of mRNA. How? Because if an mRNA is translationally active, it will be recruited heavily by the heavy ribosome, and the density will be high and then concentrate at the bottom of the gradient. And those pool of mRNA that is not bound to the mRNA and not bound to the ribosome will be light and will be floating in the top of the gradient. In this way, we can resolve, and then if you collect it through the continuous collection system and then monitor through UV, typically you get this pattern. So this part is polyribosome, a bound mRNA, and this part is silence pool of mRNA, means non-bound mRNA. Then we extract all the mRNA from different part, and then you have to remember that time it was 2009, and RNA-seq was not very popular, was not even in the market that time. So we did whatever best available that time, which was asymmetric gene chip analysis, where the whole human genome is immobilized on a chip, And then we probe that chip with the mRNA isolated from this silence pool and active pool with and without interferon gamma, and then did the polysome profile analysis. So what we are looking at here is that this is the color code. Means green means less, red means high. So now for the unfractionated mRNA, means non-resolved mRNA, we can see that interferon gamma treatment causes, so these are the genes. This is actually a partial list that time. So gene is induced, see red means most of the genes are induced. However, if you see the same gene that is induced in response to interferon gamma, but if you measure the ratio of polysomal versus non-polysomal, means polysomal mRNA versus non-polysomal mRNA, that is a quantitative measure of the translatability of a particular messenger RNA. So that's what we did here. So this is the final data. Although these messages are induced in response to interferon gamma, however, their ratio is not getting high. So the message is more, but they are not efficiently recruited to the mRNA, to the ribosome. So that means these messages could be under translation control. And as a proof of concept, we have found ceruloplasmin also here. So then, we did a lot of work that time and identifying each message separately. And then this is one of the data from that endeavor. So we deliberately knocked down L13A from the monocytes, and then did polysome non-polysome ratio, and we can see that in the control cells, the ratio of these chemokine and chemokine receptor, very important protein, takes part in the inflammatory response. So these are the protein that calls the macrophage at the site of inflammation, and then sustain inflammation. And those macrophages will make these molecules to kill the invading microorganisms. So under normal condition, these molecule expression, although the interferon gamma pumped up their messenger RNA, however, their polysomal abundance is low under control cells, means under normal condition. Now, if you remove L13A by RNAi, this is the status of removal, then you can see all of a sudden their polysomal abundance is high. So this is actually the data derived from the after the bioinformatic analysis of the Affymetrix gene chip. So we come up with a model at this time that, well, a macrophage or monocyte in response to interferon gamma produces lot of inflammatory protein. So their protein translation is going on up to 8 hours of interferon treatment. This is on the basis of our data at that time. And these molecules, protein synthesis of this molecule will generate those molecules, and then inflammation will sustain. However, this should not go for a long time. The cell must have adopted a control mechanism, and this GAIT complex may be a part of the control mechanism that L13A is released. That is happening in a delayed fashion and form GAIT complex. And once GAIT complex is made, so this GAIT complex can bind the 3' UTR. All of these elements may have the GAIT element at their 3' UTR, and then this GAIT complex can bind the element and then silence their translation. So this could be a natural defense against over-inflammation. This is the way maybe the cell is controlling the synthesis of these lethal molecules. But again, this is a hypothesis. So how to test that hypothesis? See, hypothesis doesn't make any sense if it is not testable. So we have tested the hypothesis more direct, but little bit challenging way. So by generating an F1 mouse of the conditional null allele of L13A. So this mouse only in the immune cell means macrophage, mostly macrophage and monocytes, there will be no L13A. So how we have generated, but not in other organ. How we have generated? So this is entirely generated in my lab, this model. So we actually put the LOX-P site into the genome of L13A. So this is the exon intron boundary. So we put LOX-P site, this is the LOX-P site, into the between exon 1 and 2 and then at the end. So that this genome is called technically is called floxed genome. So then we identified embryonic stem cells that is transformed by this construct, and then identified by PCR the ES cell that is harboring the conditional null allele, and then inject that ES cell into the surrogate mother, and then this guy, the chimeric mouse, was born. You can see that this mixed fur color. That means this mouse got the genomic DNA from the ES cell. This color is the marker of this construct. So, and then we have before injecting, actually we have tested the ES cell whether there is a Cre-dependent excision by infecting with adenovirus Cre, and you can see that upon infection of adenovirus Cre, the L13A expression is going down. So, and then we have generated the F1 mouse, and now the F1 mouse is only harboring the conditional null allele, means the flox genome of L13A. Then we cross this mouse with Lysem Cre mouse. What is Lysem Cre mouse? Lysem Cre is a Cre recombinase enzyme that can recognize the LOX-P site and cut it. So, but in this construct, the expression of Cre is driven by the lysosome M specific promoter. So, lysosome M specific promoter is not active in other organ, but highly active in the cells of myeloid lineage. And we made this mouse by crossing the flox mouse with the Lysem Cre mouse, and then harvested macrophage from this mouse and different organ from this mouse. And they made the lysate and then did a Western blot. So we can see that in macrophage, there is no L13A of the flox Cre mouse, but the same mouse, when we are testing liver and kidney, L13A expression is not compromised. So that shows that we have indeed generated a macrophage specific L13A knockout mouse. And we are going to use this mouse to test our hypothesis, whether what is the inflammatory status of that mouse. Is this inflammation is high? So we have used that mouse using 3 human disease model of murine. One is LPS-induced endotoxemia. That endotoxemia causes certain inflammatory burst, and people may die, and it mostly happen after the surgery. And also another model, high-fat diet-induced atherosclerosis, which is cardiovascular disease. This is also an inflammatory disease of the immune cells of the blood vessel wall. So, in crude sense, it called heart attack. What is how heart attack happen? So, mostly it happens by when the artery got clogged with the cellular debris, and that formation of that cellular debris is an inflammatory response of a process called atherosclerosis. So now we have a mouse model that can mimic atherosclerosis. We have used that model, and also we have used DSS-induced ulcerative colitis model. I do not have time today to describe this model, but I will just show you a little bit of data from these 2 models. And also, our mouse got accepted by the Jackson Laboratory, which is the world's largest mouse repository of genetically engineered mouse, as a model to study resolution of inflammation. And you can see that in the nomenclature, I didn't know that they put actually the investigator's last name here, Mazumder's, just for these 4 letters. So this mouse is available through Jackson Laboratory, and many lab has actually used this mouse, and including in drug discovery against inflammation. So what we did at the beginning using this mouse is we challenged this mouse with LPS to cause inflammation, and then test how this inflammation is resolving in the knockout mouse, in the macrophage specific L13A knockout mouse versus control mouse. And we can see that in the knockout mouse, typically we saw huge blood clot is happening in the lung. See, lung is the very prominent target organ for overinflammation. And we can see that in the bronchus, it's rapidly getting filled up with neutrophil, and also in the blood vessel of the lung, macrophage got lined up, which is the typical scenario of multi-organ failure. That's what happened. And we can see that, this is what happened in endotoxemia. And we can see the same thing in this mouse that happening in a very accelerated fashion in this knockout mouse. But so this LPS is LPS treatment is actually sublethal. It is not a lethal dose, but even with sublethal dose, the knockout mouse is dying. And we have also measured the infiltration of neutrophils and macrophage in the peritoneum by FACS, fluorescence assisted cell sorting analysis, or FACS analysis, using these two markers, GR1 CD11B, which is and F480 CD11B and F480 GR1, to assess the abundance, the relative abundance of the inflammatory cells. And we can see that in the knockout mouse, these the abundance are very high in the peritoneum. So, but what is the relationship with translation control? So, is translation control of these inflammatory molecules is compromised there? So, we also undertook a very, very challenging experiment by isolating primary macrophage from these mouse, control mouse and knockout mouse, and then resolve the ribosome, and then isolate the polyribosomal fraction, means the translationally active pool, and then determine the relative abundance of these three cytokines. We can see here, under control condition, means normal condition, normal mouse, these mRNAs are induced. However, these mRNAs are mostly in the silence part, means not very efficiently producing protein. However, when the L13A is gone in macrophage, that means macrophage specific knockout mouse, you can see these mRNA are now more in the translating, active translating fraction in the polyribosome. So their relative abundance are also very high, and that might contribute to these over-inflammation. So now we move. I'm going to move to the next model. That means high-fat DAT-induced atherosclerosis. High-fat DAT is inflammatory and causes atherosclerosis. That means it actually accelerates the formation of the atherosclerotic plaque in our blood vessel. However, in mouse model, there is a problem to study this, because mouse do not, no matter how high fat DAT you feed the mouse don't get atherosclerosis because of its very high lipid efflux pathway or high HDL pathway. But if you knock out one gene, ApoE gene, in this mouse, then this mouse, and feed this mouse with high fat DAT, then you can see rapid atherosclerosis and just mimic human atherosclerosis in mouse artery. And this mouse has been developed by Oliver Smithy, who got Nobel Prize for the generation of gene knockout mouse. So we have used that model. So, and this is a very standard pathway to use this model. So we cross ApoE with our macrophage specific knockout mouse, and then generated another chimeric. So breed this background in ApoE knockout background. And then we feed this mouse with high fat DAT for 12 weeks and retrieve the aorta, and then stain the aorta with oil redo, with actually specifically stained lipids part to see the atherosclerotic plaque. And as a control, so this mouse we have used as a control. So we breed L13A flux mouse, ApoE background as a control, and flux Cree mouse in ApoE background as a experimental mouse. So this is the control mouse, and this is the experimental mouse. Means everything is there except L13A is not there in the macrophage in this mouse. And then feed this mouse high fat DAT for 12 weeks, and then retrieve the aorta and stain the aorta with lipid oil redo specific stain. So what you are looking at is the aortic root analysis. Means the chamber of the heart that is getting rapidly filling up with atherosclerotic plaque. So we can see that in the when L13A is not there, the mouse is under high inflammation and supporting rapid filling up of the chambers by the plaque. So this is what typically happen in human also, and when it crosses the threshold, so there is plaque rupture happen, and that might cause either cerebral or myocardial infarctions. So, and very recently, we actually detected, specifically the what gene expression is altered and how gene expression is altered, specifically in the plaque that is rapidly filling up the artery. So how we did it? So this is a method called laser capture micro dissection by isolating the plaque with very high precision. And you can see that after isolation of the plaque, this looks like this. So this plaque is gone now. And we subjected this plaque with the RNA nanostring analysis to measure the gene expression on a global level. So what is RNA nanostring analysis? This is a color-coded molecular barcode based reporter probe that we can target any gene, and so this probe set you can buy, and then probe with your RNA. So we use these inflammatory probes and then probe the RNA isolated from this plaque, and then this is the final data that recently published in FASEB. So we can see that the many inflammatory gene, more than 1 or 2 log-fold expression. This is a volcano plot of a group of inflammatory genes, and are upregulated in this plaque. And then actually we have validated them with independent experiment using real-time PCR, particularly for those genes. And all of these genes, bioinformatic analysis showed that GAIT element is there. So this is how the GAIT element looks like. I will talk a little bit more on this element. But this is bioinformatic analysis from this. So, so at this stage, so again, I want to remind you the hypothesis that this could be a resolving mechanism for overinflammation. How GAIT element looks like? This is the original GAIT element that we have found in the 3' UTR of celluloplasmin message. And the energy value look like 1.2 kilocalorie per mole. So, however, the GAIT element that we have found subsequently in other target message, that these chemokines, human, CCL2, mouse, human, mouse. So you will not see any sequence homology between them. There is no sequence homology. However, there is a folding homology, and bioinformatic analysis of this folding homology was possible because of the excellent program of called Fold Align is available. So this Fold Align program will pairwise structural RNA alignments that identified a number of predicted stem loop structure in the target mRNA, where no apparent sequence homology exists. So this is actually a pretty big deal. So, and then we have, so this is a bioinformatic analysis, and bioinformatic analysis has its own limitation until and unless you actually test them or prove them independently. So we did actually identify this structure or validate this structure in a more direct way by conducting 2D NMR analysis, where the proton noisy spectra, we can see that is actually what this spectra will tell. So if a particular bonding will produce particular immunoproton resonance, and we have found the correct immunoproton resonance that is responsible for AU bond or GC bond by this 2D NMR. These are the resonance spot here, and this data actually supports that this is not only on the basis of bioinformatic analysis. In reality, these bonds actually exist on the basis of this resonance data. So now I want to remind you our hypothesis at this stage, that if you treat macrophage or monocyte with interferon gamma, it synthesizes pro-inflammatory protein. Inflammation happen, and this inflammation is necessary to protect us from infection. However, these inflammation should be controlled, and inhibition of a translation of the group of inflammatory proteins will resolve inflammation under normal condition. So this will happen through a late response of the GAIT pathway. So L13A is released by phosphorylation, bound to this multi-protein complex, forms a multi-protein RNA binding complex that binds the active GAIT element, and this binding will directly inhibit translation. So this translation inhibition depends on this binding, and then inflammation will be resolved, and no disease and maintenance of healthy state. However, if there is any defect in this state binding, or defect in L13A, or defect in the RNA component, inflammation will not be resolved, the fire will continue to burn, and then that might ended up in pathogenesis of many human disease, such as heart disease, sepsis, colitis, autoimmune disease, so many inflammatory disease. So now how you can use this GAIT element for the discovery of new generations of anti-inflammatory drugs. So this is a very new direction that is emerging from my lab. So what we have identified that this GAIT element, if you deliberately hook this GAIT element with a reporter gene, this presence of this GAIT element will also inhibit the expression of this reporter gene under right condition. And as a control, we have measured another designed another reporter gene, which is luciferase based, where there is no GAIT element. And then generate stable transfected mouse macrophage line expressing this reporter luciferase under the control of GAIT element. And then we have identified this line. So this is the luciferase expression level. And then use this cell line to treat this cell line with interferon gamma. And you can see that treatment with interferon gamma is the positive control that will induce GAIT element dependent translation silencing and inhibit the expression of the reporter gene. And you can see that there is inhibition of expression, and by analyzing the statistical data, we have measured the Z factor, which is very important for drug discovery, important for the development of a cellular platform for drug screening. So we have identified the Z factor between 0.5 to 0.9, which actually reflects the dependability of this high throughput assay. And there is a bunch of chemical libraries now under the screening platform: Cambridge, LOPAC, Spectrum, NIH clinical collections. So lot of compounds under. So this will make my lab busy for a couple of years. So so far we have screened LOPAC libraries, and you can see here that how false positives are also important in drug discovery. So and what is the importance of the control in drug discovery? So all of a sudden we found these compounds that is inhibiting the translation. However, we should not get too excited, because even without the GAIT element, these compounds are also inhibiting. That means this inhibition is just the inhibition of the luciferase. This is not mediated by GAIT element, so we are not interested. These are false positive. However, we identified two compounds. This is showing inhibition, but not showing inhibition when we are using a platform without the GAIT element. So that means these two compounds may have a potential. So this called potential hits. So in this way, we will collect all potential hits after screening of all library and then test them in animal model or using cellular model. So without going further detail, I'll just now switch to another emerging work from my lab, that how this translation control mechanism regulates the human pathogenic respiratory virus. The first virus we have, so we have published this paper a couple of years before, that extra ribosomal L13A could be a specific innate immune factor for antiviral defense. So we have used respiratory syncytial virus at that time, so which is a which infects children within the first 2 years of their life and infects the upper and lower respiratory tract of the children. And it is the clinical manifestation is bronchiolitis, pneumonia, and asthma. So, and also implicated in the sudden infant death syndrome. Until now, there is no vaccine is available for this virus, and repeated vaccine development was trial was unsuccessful. So the RSV genome looks like this. So this is the genomic RNA is a negative strand RNA. That means this RNA directly do not produce message. This is the anti-message strand RNA that's being packaged in the matured virion, and these are the viral protein encoded by the genome. N is the nucleocapsid protein, P is the phosphoprotein, M protein, S, I, G, and F. These proteins are present, are very important for infecting the cells, and L is the RNA-dependent RNA polymerase. It has 11 mRNA and 2 leader RNA. So rationale for this work is clinical trial of RSV vaccine failed repeatedly, and outcome of this disease, so this is important, the outcome of this disease was more severe when those immunized individual face the natural infection. So in a clinical trial, the children who got the vaccine, actually when actual virus came, so the children who got the vaccine in clinical trial got more severely ill. So this is dangerous. So that's why it is important to do a rigorous phase 3 clinical trial, even for this COVID vaccine also. So how this virus interacts with the component of the cellular innate immune mechanism, monocytes, macrophage, is really largely unknown. So rationale for this work is inflammation in the airway of the epithelial cell of lung. This inflammation plays a very important role in the innate immune response against RSV or against all respiratory virus. The people are dying in COVID because of the cytokine storm or rapid inflammatory syndrome that is happening, and not because of the virus per se, because of the cellular response of the virus. So our previous work identified, I showed data that ribosomal protein L13A dependent silencing mechanism is responsible for reducing inflammation. Since inflammation is actually also working against the virus, so in this system, when L13A is knocked out, inflammation will be high, and we hypothesize that maybe this mouse will be resistant to the RSV growth. But we really got hit hard by getting the opposite result of the hypothesis. So it is not necessarily true that in experiment we will prove the hypothesis. We have to prove the hypothesis. It's not at all true, and I don't believe in that. A hypothesis could be correct, could be wrong, but testing is more important. So we should look at the data with a very unbiased mind. So what we have found, look at here. So what we have found that L13A knocked down lung cell. So the RSV growth and L13A knocked down mouse macrophage, and we infect those cells with RSV, respiratory syncytial virus, and we saw in the knockdown mouse, actually the viral growth is more. This is the syncytium formation, you can see here. Viral growth is more. Why viral growth is more? So we expected that over-inflammation or inflammation will inhibit the virus, but that's not happening. So what is happening here? So we have identified that L13A could be a translational repressor of a RSV matrix protein M. This protein M that is encoded by the viral mRNA is a rate-limiting protein of the virus. So if you inhibit M by RNAi, so you can see that the viral growth is reduced. So this is showing how important M is for sustaining viral growth. So now we have infected the cells with this virus, both the control cell and the knockout cell, and measured polysome, non-polysome ratio of each viral mRNA. And here I will draw your attention to this. The M gene of this RSV virus in the knockout cell, they are more recruited into the polysome. Means when L13 is not there, these messenger RNA translation is high. And that is also, we have tested by independent experiment. This is the polysome analysis. We can see that the virus infected cell, control cell, M protein is less in the polyribosome fraction. That means the translationally active pool. But in the knockout cell, M protein is actually more in the polysome. That means when L13 is not there, M protein synthesis is going high, and that may stimulate the viral growth. Because under normal condition, virus itself do not allowing the M protein to grow high. Because why? This is also a $1 million question, because it is pretty much possible that virus do not want to grow very high rate, because then it might kill the host, and then it may cause a detrimental effect of the then virus even cannot grow. So virus has a pressure always not to grow robust, so that to keep the host alive. And in the 3' UTR of the M gene mRNA, we have identified actually GAIT element with fold align, because there is no sequence homology, fold align, and then design this construct, transfer this construct into the cell and hit the cell with RSV. And you can see that under RSV treated condition, the construct harboring the GAIT element from the M gene, actually expression is less. So we have identified a virus activated inhibitor of translation. And not only for RSV, so we have seen this element in human parainfluenza virus, mouse pneumonia virus, and also Ebola virus, which these GAIT elements. And not only that, surprisingly, when we are working on this, after a couple of years, all of a sudden, a group from Spain published a paper where they have identified GAIT element into the gastroenteritis coronavirus genome that might modulating innate immune response. So this is not our work. This is the work from this group in Spain. So this is the first GAIT-like motif described in a positive RNA virus. So they have identified this in gastroenteritis coronavirus genome, which is a near relative of the SARS-CoV-2. So the next question we ask, can GAIT element regulate SARS-CoV-2? So this is the genomic organization of SARS-CoV-2. So many of you might have know this by this time, because a lot of people are talking about this. The SARS-CoV-2 genome has about 30,000 RNA in its genome as a genomic material. The SARS-CoV-2 infect the cell. The first job it is doing is translating ORF1ab protein. So ORF1ab protein is being made from the genome, and then this protein synthesized as a big polyprotein. But the protein is dead until and unless a viral protease actually chop the protein into the 16 different pieces. And each piece played an enormous role in the viral replication inside the cell, in the infected cell. However, none of the protein got packaged into the mature virion. That's why all of them called non-structural protein. And the heart of this virus is RNA-dependent RNA polymerase that is also being encoded by one of these protein called NSP12. That is directly coming from ORF1ab. And spike protein, E, M, and N protein, these are the structural proteins. Spike protein, as you know, that this is the largest structural protein of SARS-CoV-2. This is a glycoprotein and essential for entry into the host cell. And this protein is actually the target antigen for most of the vaccines now currently being made. And there is another protein called E protein, envelope protein, membrane matrix protein, and nucleocapsid protein. This protein actually wrap the genomic material and protect the material. So all of these proteins are structural protein. So this is the life cycle of this virus. So, this virus infects cell using the ACE2 and TMP RSS2. So this is the receptor that is causing opening the gate of the virus by interaction of ACE protein with the ACE2 receptor, angiotensin converting 2 receptor. So then the virus get entry and eject the genomic material. So first thing the genomic material do is make the polyprotein from the ORF1ab, and the viral protease 3CL-Pro cut the protein and generates different pieces. Many of these pieces are also required to confuse the immune cell, so that the virus replication can sustain. And one of the protein from this piece is NSP12, which is the RNA-dependent RNA polymerase. As soon as this protein been made, this protein will copy the viral genome and make the message strand, then copy it again. So replication, transcription, this is going on. This will start, and then all of the messenger RNA will be made. That means messenger RNA of S, E, M, N protein, and this protein will be translated and immediately go through the ER Golgi pathway. So it will actually hijack the ER Golgi pathway of the host cell and then send those protein right to the membrane and align themselves in the membrane so that the baby virus, when it will be ejected from the cell, will get this membrane assigned protein right away and then equip itself with again S protein and ready to infect another cell. So this S protein, how this S protein goes there and how the S protein translations is happening is a very critical step of the viral pathogenesis. So we ask the question: is this translation is under any sort of control? So is this kind of element exist in the S mRNA? So we have no idea at that point. So, and in this lockdown, first 1 month, my lab was totally shut down and we have nothing to do, and then was using this Wuhan strain of SARS-CoV-2 genome to look for whether any potential gate element exists, and we got hit right away. So ORF1ab, and we have found 3 gate element in the SARS-CoV-2 smRNA, which has a folding homology but not sequence homology, which is the typical characteristics. But is it real gate? This is again bioinformatic analysis. It doesn't make any sense unless we could test this. So is this S protein is sort of any sort of translation control? So that's the question we'll be asking now. And before that, I want to tell you that so when virus interacts with cellular receptor ACE2 through the viral S protein, it causes opening of this gate. But is this the only job of this interaction of this, that entry of the virus? Maybe not, because one group published number of years before from Hong Kong at the time of SARS-CoV infection, when SARS-CoV infection came, not CoV-2, the previous SARS. So they have identified that S protein interaction with ACE2 triggered a signal transduction pathway that activates casein kinase 2 and activates RAS, RAF, MEK, and ARC pathway, and ultimately signals the nucleus, and that activates CCL2 mRNA transcriptional induction of CCL2 mRNA. However, we have identified that this pathway may have a crosstalk with the pathway for L13F phosphorylation. That means L13F phosphorylation required activation of a signal transaction pathway involving DAP kinase and ZIP kinase. So I'm not going to that episode. That's a huge saga. I don't have time to cover that. So we found a crosstalk between that pathway and this pathway. So immediately we asked, so if that is true, then can we do some kind of initial experiment using a recombinant system of S protein and human lung cell? So immediately we have generated this construct, that's reporter construct, where the gate element under the control of COV2S mRNA and ORF1ab mRNA, the one that we have identified in this bioinformatic analysis. So we have generated this construct, and this is the without gate element control RNA, which must be there at the same time. And then in vitro transcribe this in order to generate the chimeric reporter RNA with the luciferase and the gate element. And then in vitro translate this chimeric construct in rabbit reticulocyte lysate in presence of S35 methionine to see the de novo translation, means new translation from this exogenously added mRNA harboring the gate element from SARS-CoV-2. And in this system, we can add external influencer with that what can inhibit this translation, gate element mediated translation. So we have used human primary bronchial epithelial cells and treat that cell with SARS-CoV-2 S protein to ask whether it can activate this pathway, and tested and whether that activation could lead to the translation silencing, and tested this extract by adding this extract to this in vitro translation system, and then run the SDS page where the intensity of the de novo synthesized reporter protein, luciferase protein, will directly reflect the efficiency of translation. If it inhibits, then inhibits under the gate element or not. And now I'll show you the data that extract made from cells treated with S1 subunit of SARS-CoV-2 S protein, and this is the A459 human lung epithelial cell, and this is the primary lung epithelial cell from human. And both the cases, we can see that if you treat the cell with S protein, we can activate an activity that is actually inhibiting the reporter gene under the control of the gate element. This, but not the reporter gene which is not under the control. So this, in this system, we have actually co-translating this RNA also without the gate element. And we can see that this without the gate element translation is not subject to the inhibition of the inhibited extract, but only the reporter gene with the gate element is sensitive to this extract. And we can repeat that experiment using another human lung cell line A549 cell. But most interestingly, this activity we can turn on only by treatment with CoV2S protein, but not with interferon gamma. So, because interferon gamma, if you treat interferon gamma, we cannot inhibit translation. So that means, although it is a gate element dependent activity, but it is not, for viral context, it is not turning on by interferon gamma. So as we know, and then we did some mechanistic experiment also. Is the silencing competent lung cell extract, means this extract can bind the RNA binding protein to the RNA, because gate element mediated translational silencing requires the RNA binding protein should directly bind the RNA. Is this binding is happening? So this is the silencing competent lung cell extract made from the spike protein treated cells can support RNA binding complex. So this is a RNA gel shift assay. We are using the gate element from this as a probe, means gate element from this as a probe, and then incubate this radioactively labeled probe with the cell extract made from either S protein, SARS-CoV-2 S protein treated lung cell, or nothing treated lung cell. And then we can see in the non-treated cell, there is no RNA binding complex, but we can see a successful RNA binding complex formation only with the extract that has been treated with SARS-CoV-2 S1 subunit. And this RNA binding complex can be competed out with the competitor RNA, same self-competitor RNA, means the cold RNA without radioactivity, means 10-fold or 20-fold excess, molar excess, and that can compete, which shows the specificity of the binding. But we agree that this experiment is actually now raising more questions than answer, because this is a very new project. We didn't even talk about it. This is the first time I'm talking about it here. So why many virus has adopted inhibitory elements in its genome? Why a virus would adopt inhibitory element? So if this is a weakness, there may be a reason. Maybe virus don't want to replicate high, so that high replication might kill the host cell immediately, and that might compromise the growth of the virus. So their virus is always experiencing a selection pressure not to grow high. So if this is a weakness of the virus, then can it be exploited as a target for novel antiviral drug screening? So can we use the same cellular platform that we have showed to screen novel antiviral drug, which can deliberately promote the GAIT element mediated silencing to counter the virus growth? What are the signaling mechanism activate the silencing? So what are the identity of the RNA binding protein? So what is this protein? Is L13A there? And what is the identity of this protein? We don't know. So that will be that will also make my lab busy in the coming days. What are the identity of the RNA binding protein? Other players and cellular factors. But finally, we have to recapitulate this event in live SARS-CoV-2 infection model and reverse genetics based virus model using engineered translation regulatory elements in the virus. So we have to create synthetic virus, which by manipulating the GAIT element, and then see whether that affects the virus growth. So those experiments are also in the pipeline. So I will stop here and acknowledge the students. I didn't do any of the experiment by myself. All of my students have done it. So this is my group here, and these are my collaborators, especially Anton Komar, which is an excellent collaborator. My next door neighbor in my department is a protein biochemist. So William Baldwin from Cleveland Clinic. Balton Tolbert, who helped me in the 2D NMR from Case Western Reserve RNA Center. Doug Lehman, who helped the Affymetrix gene chip experiment. Maria Febraio, Jonathan Smith in the mouse experiment, and Bill Merrick, who is a translation control guru from the Case Western Reserve University, who actually identified or invented many of the translation initiation factors. And Mark Cameron for bioinformatic analysis, and funding from National Heart, Lung and Blood Institute of NIH. I was lucky enough to get the major R01 grant from the 2004, which is now experiencing like fifth or fourth, competing renewal. American Heart Association was. This is a critical point in my career by getting the scientist development grant from them, which helped me in the early stage of my career. Also the grant in aid from American Heart Association. Many of my pre-doctoral and postdoctoral fellows in my lab who worked also got grant from American Heart Association and Center for Gene Regulation in Health and Disease from our university for their excellent support for every step of this work. So I will stop here. I just want to tell you that apart from my work as a biologist or as an immunologist or molecular biologist, I have another hobby of writing Bengali poetry, and these are some of my publications in that front also. So I will stop here and entertain any questions. Thank you for your attention.
Thank you, Professor Mazumder, for such an insightful talk spanning inflammation and the role of these regulatory complexes, along with your recent insights on how this can be exploited to understand the current pandemic. And we are also privileged to know about the last slide on your hobby on this book. So that's really an advantage to have these things in this platform. So we have received many questions from participants attending both from Google Meet and YouTube. And if I may ask you a couple of them, and if you don't mind answering them.
Sure, I'll try my best. I don't know whether.
So we'll start with a question by Gaurav Bharadwaj and Ratan Gachui on L13A. Here's a question on which protein phosphorylate L13A and what is its phylogenetic presence, like whether it's present in all the three domains of life, like archaeobacteria and eukaryotes.
No, it is not present in all phylogenetic domain of life. So this is a very specific for mammalian cell in higher mammals and especially in the immune cells. And there is a big story on what phosphorylates L13A published in Molecular Cell. I'm not really went to that, and that's the work was published from Paul Fox's lab in Cleveland Clinic. So they have identified a cascade. I think this work was done by Rupak Mukhopadhyay when he was working in Paul Fox's lab. So they have identified a cascade like DAP kinase and ZIP kinase that is responsible for L13A phosphorylation. But whether the same cascade is working in virus, we don't know. We don't even know whether L13A is playing any role in virus. So we didn't do any knockout experiment in virus. So those are upcoming. But for L13A phosphorylation, yes, the entire signal transaction pathway has been dissected and the players are known.
Okay, thank you. So there is a question by Sandeep Pramanik. Is there any differences between L13A and ApoE genes?
Yeah, those are two different genes. ApoE genes are responsible for lipid metabolism. You know, lipids are not, mostly the lipids are floating in lipoprotein complex. So ApoE is the protein component. LDL, so the low density lipoprotein, ApoE is the protein component. So if we knock down ApoE, then it cannot be cleared by the receptor mediated endocytosis pathway, and that's why it accumulates lipid and causes disease. So that is essential to induce disease, human atherosclerosis in mouse, by making them ApoE knockout. But L13A is another different protein. L13A is a protein of the large subunit of ribosome, we have identified a new function of L13A that can be released in response to interferon gamma treatment and causes gene-specific translational silencing. And yes, L13A and ApoE are two different proteins with entirely two different functions.
Okay. And there's a question by Bishwadip Ghosroy. What is the correlation between L13A and Treg cells?
L13A and what cell?
Treg, regulatory T cells.
Oh, regulatory T cells. We have not pursued that avenue, but we know that mostly L13A is a ribosomal protein, so it will be there in all cells. All cells need ribosome to synthesize protein, right? And all ribosomes has L13A. So no matter what kind of cells, this protein is present, in all of our cells. And what is the function of this protein they have a generic function. Ribosome function is generic, like protein synthesis. So that is generic in all cells. But whether apart from the generic activity, whether different cells can use this protein in different way, all knowledge is not known. So far what we know, that in immune cells, especially in monocytes and macrophages, these protein plays, apart from its role in ribosome, these protein plays a very critical role in translation control by gene silencing, directly translation silencing.
Okay. There is a question by Deepak Korea. Does L13A, when on the ATS ribosome, play any role in general translation? And if so, then how does cell cope up with the L13A knockout?
Yes, very, very important question. Very, I like that somebody asked this question. There is always, this is the reason why we face very hard time when we created a L13A knock. Before creating the mouse, one of my postdocs, Sujan Choudhury in my lab, he actually generated L13A knockdown cells, where by RNAi, where there is no L13A. However, proliferation, protein synthesis of that cells remain same with the wild type cell. So that directly shows that these protein, although this is a ribosomal protein, but these protein do not take part in directly into the protein synthesis activity of the ribosome. But this is true only for mammalian cells. But in lower eukaryotes, yeast, if you knock down L13, then we get a lethal phenotype. But not in mammalian. So we believe that this activity of this protein may have evolved, when as the cell become more complex from yeast to mammal.
Right. So there is another follow-up question by Deepak. The L13A fluxed mouse model gave excellent insights into the gate-regulated translation. However, the L13A locus also encodes several SNO RNA. How much contributions of the loss of SNO RNAs you think might have in the flux?
Yeah, I think somebody worked on that, but we found, I didn't show that data because of the lack of time. I don't think that there may be some compromise of the SNO RNA there. Yes, there are some SNO RNA locus in that genome, but in the knockout mouse, in the macrophage specific knockout mouse, we didn't see any defect of the mouse if we do not challenge the mouse with inflammatory stimuli. Under normal condition, the wild type mouse and the knockout mouse looks same. There is no difference. They are breeding competent. They are happy. They live almost, their lifetime is also same. So there is no difference. But we see the difference when we challenge the mouse with inflammatory stimuli, either with LPS, either with high-fat diet, either with DSS. Then we see the knockout mouse is falling apart very rapidly, but not the wild type mouse.
Okay. So I think we can move from L13A, and there's a question by Shubhomit Laha. How is chemokine CCL2 linked to inflammation and cancer pathogenesis?
I don't work on cancer, but cancer has a big relation with chemokine because again, inflammation is also a double-edged sword for cancer. So inflammation stimulates cancer, and also inflammation can work against cancer also, because cytotoxic T cell can kill the neoplastic cancer cells. But CCL2 is actually the macrophage homing factor. So it actually it is showing the macrophage where to come. It is like a light for the macrophage, and macrophage follows that chemokine gradient to see where is the site of inflammation. So in the site of inflammation, the cells secrete those things, and then more macrophage follows that gradient and to come to the site for inflammation. So this kind of mechanism may be important for metastasis also. So inflammation could be pro-metastatic also. So there are a lot of papers on that. So
okay.
Yes, it is a double-edged sword.
So the question by Topobroto Pramanik, inflammatory responses due to cytokine storm is a major cause for comorbidity in COVID infections.
Yes.
Can somehow L13A be upregulated in human body to prevent the upregulation of the inflammatory chemokines?
If L13A under normal condition, this is the normal defense mechanism. So this is why you cannot deliberate upregulate L13A. We have to use small molecule. So that's the reason. So we need a screening system to test this hypothesis whether deliberate promotion or activation of L13A may reduce this inflammation. My gut feeling is it is happening on the stage of, if at all L13A is involved. We don't know what is happening in SARS-CoV-2 yet, but if at all L13A is involved there, so we think that we have to go for designing small molecule that can promote the release of L13A. L13A is there in all the time, in all the cell, because this is a sort of generic ribosomal protein. But its activity requires the release. So maybe the control will be done by activation or inhibition of the kinase, but not by the L13A itself. That will promote the release. If it can promote the release, then it can silence more. So those hypotheses needs to be tested in the viral infection model. Yes.
Right. Okay. So we received many questions, and we do apologize to the participants that we are not able to answer all of them. But I'll take two more questions, Professor Mazumder, if it is okay with you.
Yes. Yes.
Okay. So you got a question by Shraddha Mahendra. Can we perform, I think, genome-wide sequencing for identification of long non-coding RNA, or we should go with RNA sequencing?
RNA sequencing for what?
Identification of long non-coding RNA.
Yeah, long non-coding RNA can be explored with nowadays with directly with RNA-seq. Yes, RNA-seq will directly detect long non-coding RNA here.
Okay, and we'll take the last question by Sujan Chaudhary. How VAIT is different than GAIT?
How weight is different than GAIT? Yes.
Yeah.
Is Sujan Chaudhary? Is Sujan? I know, who worked in my lab.
That we are not sure.
Okay.
Probably.
Because there is one Sujan Chaudhary who actually worked in my lab as a postdoc and did a very good job on RNA knockout
He says
cells.
yes. This is probably the same one.
Okay, nice to know. So the weight is, we don't know whether these two are same, but I don't think these two are absolutely same. Maybe there is a part homology exists between weight and GAIT, but the main difference between weight and GAIT is weight has nothing to do with interferon gamma, because interferon gamma treatment cannot activate viral translational silencing. Only the virus can, but not by not interferon gamma. Both of them could be L13A dependent, but there is substantial difference exists between these two. Yes.
Okay. I think we'll end with this last question by Dr. Arja Mitra, but I think you have explained this during your talk, is if you tell something about the genetically engineered animal model developed by your lab on the RPL13A knockout mice, conditional knockout mice, which is now used by many labs to study inflammation.
Yes, yes, and I have no control on the distribution now. Now we have outsourced. Now actually Jackson Lab is doing this. So if anybody want that mouse, so that is actually a flox mouse. And according to the question of the investigator, if he wants to make another tissue, say liver specific knockout mouse, then he or she has to use a liver specific Cree, not lysine Cree, a liver specific Cree or another lung. If lung specific L13A knockout, we are also doing this with Clara cell specific L13A knockout mouse using CCT Cree. So by crossing this mouse. So yes, it depends on what kind of tissue specific knockout mouse the investigator wants to generate or build.
Okay. With that, now thank you, Professor Mazumder, for taking the time to speak in a webinar series and for answering so many questions. And we do apologize to the participants for not able to cover all of them on the count of time. But thank you once again for joining, and thank you to Professor Mazumder.
So thank you. It is really a pleasure for me to give a talk and then address some of the questions.
With that note, I'll now request Professor Inara Banerjee to express the vote of thanks and close today's session. Thank you.
Thank you, Shaunak. As usual, you have conducted it so nicely, and thank you, Barsanjit Babu. আপনি এত চমৎকার বললেন, you spoke wonderfully, and literally আমি প্রত্যেকটা শব্দ গিললাম আপনার, কারণ there is an overlap.
Thank you so much.
For the non-Bengalis here in this platform, and you have a few hundred people watching you on both channels, and I literally hung on to every word, because in the previous week what I spoke, I see a lot of overlap, so we will have to take our discussion offline. And when I was introducing you, I thought of saying about the poet side of you. So I'm so glad that you could also share some of the books that you have published. So wonderful work, and wish you all the best.
Thank you so much. It is really a pleasure from my end too. Thank you.
And Srimoti Di has made a request for the Bengali audience, and I am a member of the West Bengal Council of Science and Technology, and we are about to start a webinar series. So I will use this platform to ask Professor Mazumder that we must schedule a talk in Bengali by you. So Srimoti, your request is also going to be kept.
Thank you, Dr. Rena and Barsanjit.
Thank you, everyone. Okay. It's been a pleasure.
Thanks a lot.
Okay, great.
Okay.
Have a nice day, everyone. Bye-bye.
Okay. Bye, everybody.
Bye.